Thank you for the invitation to meet with you today and describe some of the work that we're doing in our Collaborative Research Center, known as the MECFS Research Center for Solutions for MECFS. It's quite a mouthful, but it's a disorder that deserves that and much, much more. Our center is interinstitutional. We include investigators across the country. In the basic science group, highlighted in red, you see Tony Komaroff at Harvard, John Greeley at Albert Einstein, Oliver Fien at UC Davis, who had these various programs. We also have colleagues at the National Institute of Neurological Diseases and Stroke, Avi Nath and Stephen Jacobson, and our clinical team, Susan Levine, Cindy Bateman, Dan Peterson, and Keegan Monaghetti. This is just the tip of the iceberg. Each one of us works with at least another 5 to 10 people. So it's a very large team that supports the work that I'll be describing today. The focus of our center is really on the role of infection and altered immunity in the pathogenesis of MECFS. We have focused most of our work in the Center for Infection and Immunity at Columbia University for decades on trying to understand the underpinnings of various acute and chronic disorders that we believe lie in infection. The rationale for this application in MECFS is that 80% of the patients with MECFS who've been documented exhaustively report some sort of an influenza-like illness in the weeks or months prior to the onset of MECFS. We also know that people who've had acute infections that have been well documented can develop a disorder that looks like MECFS, and there are reports that after Epstein-Barr virus infections worldwide, Ross River virus infections, typically in Australia, or infections with tick-borne agents develop MECFS. Our strategy has been to survey whatever can be accessed without being invasive or unduly invasive for evidence of infection. We do this in 2 ways. We use molecular assays that are very sensitive that we've built here for applications in pandemic response to look at blood, feces, and saliva for bacteria, viruses, and fungi. We also look at blood for indirect evidence of exposure to these same agents by detecting antibodies. These again are done using state-of-the-art techniques that include not only chips but also phage display. Probably beyond the scope of what I'm going to be describing today, but I will tell you about this in more detail as we have additional installments in this program. We're also interested in understanding derangements in metabolism that may underlie symptoms, and these may also give us an opportunity to identify biomarkers for diagnosis and prognosis. This is extremely important in its own right, because when we began doing this kind of work several years ago under the auspices of the Hutchins Family Foundation, people were essentially discounted, told that what they had was something that was psychological. Metabolomic findings, we were able to generate objective data that was irrefutable, that allowed the medical establishment to take patients more seriously. We survey plasma, which is a component of blood, for metabolites and proteins that differ in patients and controls. And our objective ultimately is to develop new ways in which we can treat patients based on these insights from basic research. We're also trying to bring out, to emphasize the abnormalities that we see in patients with MECFS, with the idea that the stress of exercise will amplify the abnormalities that makes it easier for us to detect and understand them, and potentially to find ways in which we can also explore intervention. We exercise patients and controls on stationary bicycles. We collect blood before and after exercise for profiling, and then we examine the impact of exercise and metabolism and proteomics. Another thing that we do, which is truly unique, is that we look at the impact of various types of stimuli in white blood cells before and after they've been exposed to microbial stimuli. And I'll show you an example of this in the True Culture system in the coming slides. We're also very interested in identifying variants of MECFS that may differ in their causes, and they may also differ in the methods for diagnosis, prevention, and treatment. The idea here is that we're going to use the database that we've built based on signs, symptoms, and laboratory findings, apply artificial intelligence that will allow us to find similarities between subjects that may link not only to diagnosis, but also to solutions for their problem. We are also building, in collaboration with others, an app that can be used on phones for collection of symptom data in real time that can alert clinicians and patients to when samples should be collected and how they might link phenomena in their daily lives to exacerbations or remissions from disease. I'll begin by talking about the microbiology. I'm referring here to what's known as the microbiome. We began looking at feces, saliva, and blood several years ago. All I have an opportunity to do today is to highlight some of the key findings. One of our first observations was that individuals of MECFS have a total amount of bacteria in their feces, which is higher than in controls. So you can see here on the left side of the screen, the average value, the number of bacterial copies that are present after PCR in a patient with MECFS versus a control. And in this instance, those findings are significant. Curiously, although the total amount of bacteria are increased in patients with MECFS, the numbers of butyrate producing bacteria are decreased. Now, what are butyrate producing bacteria? Butyrate producing bacteria are bacteria who have the ability through metabolic pathways of producing this short chain fatty acid, which is in fact what butyrate is. And this is important in controlling immune responses, among other things. So on the right side of the slide, you can see all the things that butyrate is involved in doing. It is anti-inflammatory. It's important in controlling the replication of bacterial and viral pathogens. It supports motility within the gut, and it provides a control for barrier function. So that if butyrate is in short supply, the barrier of the gastrointestinal tract may be breached, which can lead to chemicals that are normally restricted from moving into the circulation, where they can actually cross into the brain, into muscles, and cause disease. So again, on the left side of the slide, you can see that levels of butyrate are dramatically reduced in patients with MECFS. And on the right side, you can see some of the ways in which butyrate is important for contributing to normal physiology. The implications here are that if in fact butyrate can be implicated in some of the symptoms associated with MECFS, it is possible that by using probiotics or prebiotics, probiotics being the bacteria themselves, prebiotics being the nutritional sustenance for bacteria, you may be able to have an impact on disease. In fact, it's probably much easier to envision using prebiotics because they're very inexpensive and they're basically innocuous. But until we actually do a controlled double-blind trial, we won't know whether or not this has value or doesn't. This is one of the things that we hope to do in the second cycle of this MECFS Center. Tony Komarov and some of our biostatisticians have been using some of the inventories of symptoms that patients have generated for us. This is a description of the multidimensional fatigue inventory, where we look at whether or not there are correlations between the presence of specific bacterial species in feces and various complaints. Because if our hypothesis is correct that these bacteria contribute in some way to disease, that we might see differences. And the evidence here is at least there appears to be an association. Whether this is cause and effect or not remains to be seen, but it's very interesting and very encouraging that we have already found such relationships. We've done proteomic studies of plasma, and the first one, which was reported in July of 2020, demonstrated the presence of an expansion of specific B cells. B cells are cells that produce antibodies. In this era of COVID, most people now know what I'm talking about. What I want to drive home here is the point that there are several lines of evidence suggesting that patients with MECFS have an expansion of specific B cell populations that suggests that they may be responding to a particular trigger. That could be some sort of a protein that's made by us ourselves, or more likely, something that's associated with infection. It was gratifying approximately a year later to see that our findings were confirmed. And whenever you see two different groups using different methods finding the same thing, it gives us greater confidence that what we've identified is likely to be important. The implications clinically is that we can find and eliminate the triggers for this B cell clonal expansion, that we may be able to reduce symptoms. That is to say, we can mitigate disease. At the beginning of the talk, I described using exercise stress on a bicycle as a way of bringing to the fore any sort of abnormalities that we might then characterize. This is a system called True Culture, which allows us to take blood directly out of a patient's circulation and then to place it in conjunction with B cells that are also exposed to different sorts of extracts, viral extracts, bacterial extracts, various types of stimuli, and ask whether or not we can account for some of the symptoms associated with MECFS by differences that we see in patients who have disease or have controls where their B cells are exposed to these sorts of stimuli. I'm just going to show you a representative sample of some of our findings, but what I'm going to be describing is actually fairly representative of a wide range of cytokines that are abnormally responsive in individuals with MECFS. I'll begin by talking about sex-specific differences. As many of you will know, this disorder, MECFS, is more common in women than in men. And we find here that if we look at women with MECFS shown on the left, so you have the cases shown in orange and sort of an amber color versus the controls, which you see in a blue and a gray, there is a much higher level of response in the form of interleukin-6 in individuals who have MECFS as opposed to controls. We've looked here at a comparison of blood samples taken before the exercise test and after the exercise test. So that is the orange color versus the amber color. And you can see that they are very, very similar before or after exercise. Please bear this in mind because what I'm going to show you with metabolomics is quite different. And here we see the same sort of pattern in women who are the control group who are matched for age and sex and socioeconomic status, much lower levels, but no dramatic difference before or after exercise. We see the same sort of pattern in men. They are higher in individuals with disease than in the controls, but much lower than what we observe in women. These same findings can be described with a wide range of other pro-inflammatory cytokines. We're using, again, IL-6 as an example of what we see with other pro-inflammatory cytokines. We then decided to look at whether or not age had an impact. And what we found was that women over the age of 45 had much higher levels of cytokines than women less than 45 years of age. And again, there was no dramatic difference between women who participated in this particular study before or after exercise. There is a difference here that appears to be important, but in fact, it is not statistically significant. Now, the obvious question when you see this sort of a difference that can be ascribed to age is whether or not this reflects menopause. So what we then decided to do was to look at estrogen levels. And here what you can find is that in women less than 45 years of age versus women over 45 years of age, you have this difference in terms of levels of pro-inflammatory cytokines. So the impact of estrogens on inflammation and the immune system more generally are very complicated. And we've engaged with other colleagues who are experts in this area at Johns Hopkins, who will be working with us to try to dissect these. What we've described here with IL-6, this pro-inflammatory cytokine highlighted on the previous slide, is also seen with other chemokines and cytokines associated with inflammation that I've listed here in italics. Now, in contrast to what I showed you with the immune system, where we didn't see a dramatic change before and after exercise, when we examine metabolites associated with these kinds of disturbances, we find a different pattern entirely. This is a cycle known as the tricarboxylic acid cycle or the Krebs cycle, in which we generate ATP for our muscles, for our brains, and so forth. And it's an indication of mitochondrial damage. Now, what we find in individuals who are cases versus controls is that the cases have a dramatic increase in this compound known as citric acid. In the controls conversely, what we see is that levels of citric acid drop. Now, why is this important? Let me take you through the citric acid, the tricarboxylic acid cycle here. You can see here that there's this compound known as citrate, the same compound that we're measuring here. If there is a block in this TCA cycle, this particular intermediate accumulates and is released and is detected in the blood. So what we believe has happened here is that there's a block in the TCA cycle as a function of damage to the mitochondria and oxidative stress, which can be caused by inflammation, leading to, in this case, individuals with MECFS having higher levels of citrate because this doesn't go all the way around the circle and instead is released. In contrast, when you look at the controls, in response to exercise, they have the ability to crank up that cycle so that the levels of citrate are actually reduced because each crank of the cycle uses more citrate. So this is a very important finding, which if replicated, will give us insights into mitochondrial dysfunction, and it may actually lead to specific treatment modalities. Our working model at present is for understanding the pathogenesis of MECFS is that individuals who have some sort of perhaps a genetic predisposition as a result of fatigue, injury, or immunological stressors that might be caused by bacteria, viruses, or fungi have problems which occur at the level of the immune system and at the level of mitochondria that result in expression of these abnormalities that we perceive as MECFS. And that by understanding this pathophysiology and rigorously dissecting it, we will come up with ways in which we can more readily diagnose disease and we can address it with specific treatments. Now, how might this all come together in a single system? We all know about genetics because we hear about this in great detail. What people talk about less is the concept of epigenetics, which is the way in which the environment modifies the expression of the genome. So this can occur at the level of messenger RNA. These are small RNAs that have the ability to modify the expression of messenger RNAs. And these are large RNAs that have that same sort of aspect. So by interfering with the ability of RNA to bind to components of the cell known as ribosomes, where they make proteins, we have abnormalities. Now, as a result of these epigenetic perturbations, you can have autonomic nervous system dysfunction, which can result in problems with maintaining blood pressure and leading to, for example, syndrome, things like Pott's disease. You can have central nervous system abnormalities, which are perceived as brain fog. And you can also have involvement of peripheral nerves and muscle, which result in pain. The same sorts of abnormalities in the mitochondria can result in fatigue, both physical and cognitive, and can result in a prolonged recovery time. Now, the indirect effects which mediate these may be cytokines, they may be the microbiome, they may be other factors that are yet to be identified and resolved. We have one remaining year in the first cycle of funding for the center. We're cautiously optimistic that we will receive a second funding in a second cycle. But if we are successful in doing so, these are the sorts of things that we intend to pursue. We are going to be expanding beyond the use of direct molecular methods for detecting infections to use antibody tests to examine the possibility that the infectious agent may no longer be present, thinking in terms of some sort of a hit and run model where the damage is present. And the question then becomes, how do you go back historically and find what in fact was responsible for that original damage? This could result in vaccines to prevent infections and even drugs to modify the course of disease. We want to use genetic assays to look for markers that give us insights into people who are at risk for disease so that we can intervene more rapidly and more early. We want to begin to build animal models based on the findings that we have It's important to use animal models to test potential interventions, not only for safety, but also for efficacy. And we're also interested in looking at whether or not there's an overlap in the biology of individuals with some forms of long COVID and Gulf War illness that may illuminate pathways to cures. Now, people who have recovered from COVID have abnormalities that may be due to metabolic disturbances, immune disturbances. It's important that we differentiate those individuals who have structural damage, which could explain fatigue, such as lung damage, heart damage, which is very different than what we see, of course, in MECFS. It is nonetheless important and instructive to examine similarities between these syndromes. So with MECFS, as everyone on this call will know, we have an group of individuals who have a prodrome, which may consist of a sore throat, swollen lymph nodes, complaints referable to the gastrointestinal tract, allergies and hypersensitivities, sleep disturbances, fatigue, headache, and of course, cognitive dysfunction and autonomic dysfunction. In individuals who have had COVID, we know we also have this loss of smell and taste. This is not something that we see in MECFS, but we do see individuals who overlap in terms of cognitive dysfunction, palpitations, fatigue, and headache. There's another disorder that's probably less familiar to all of you, known as Gulf War illness. These are individuals who came through the first Gulf War in Iraq and Kuwait. These individuals received, in many instances, a variety of drugs which had activities on the nervous system. These were administered as antidotes for potential exposures to neurotoxins. Many of them also received many vaccines in a very, very rapid fashion over just a few days. And these individuals also have some overlap. So again, by studying these individually and in conjunction with one another, we hope to be able to get insights that will be helpful for treating veterans with Gulf War illness, people who've recovered from COVID, as well as patients with MECFS. We like to think that we are closing in on opportunities for clinical trials based on what I showed you earlier about microbiome from the microbiome research. We believe that prebiotics and perhaps probiotics that would result in increased concentrations of butyrate producing bacteria in the gastrointestinal tract might be useful. We also have evidence that there is at least a subset of individuals who have evidence of activation of herpes viruses who might be candidates for use of antivirals. And as we understand more about metabolic pathways, it may be possible to use drugs that modify metabolic pathways, or even some of the more recent CRISPR technologies, to find ways in which we can modify those as well. When we began this work several years ago, we thought that this would be something that would resolve more quickly. But frankly, the resources have been difficult to find. By resources, I mean not only funding to do the work, but encouraging scientists who can contribute their knowledge, insights, and efforts on behalf of patients with MECFS, and of course, the collection of biological materials. We have been slow to some extent, as you might imagine, over the past 2 years by the COVID pandemic, but we are encouraged and we are working as rapidly as we can on your behalf to come up with solutions that we think will be helpful in mitigating this devastating illness. Thank you very much for your attention, and we look forward to working with you in the months and years to come.
Hello, everyone. My name is Rochelle Jocelyn, and I am the Community Advisory Committee Representative for the NIH Centers for MECFS. And I'm here this morning with several researchers from the Columbia Group to pose some questions that were generated by the Community Advisory Committee. So first, would you all like to introduce yourselves?
Thank you very much for the invitation to speak to you and the community. I want to emphasize at the beginning that although everybody on this call is from Columbia, we have a large group across the United States, ranging from California and Nevada up to Massachusetts. So this is a small sampling of the people who are contributing to the work that you'll see today. I'm Ian Lipkin. I'm director of the Center for Infection Immunity at Columbia in the School of Public Health, and for the Center for Solutions for MECFS. I'm a physician, but I've spent most of my career in basic science, trained as a neurologist as well as an internist and a molecular microbiologist.
I'm Brent Williams. I'm an assistant professor in the Center for Infection and Immunity at Columbia and in the Department of Epidemiology. And my background is in molecular microbiology and specifically focusing on the role of the microbiome in human health.
Hi, my name is Che Xiaoyu, or you can refer me as Jason. I'm assistant professor in biostatistics at the Center for Infection and Immunity at Columbia University. I have a PhD in mathematics. I joined CII in 2013, focusing on research in autism and MECFS. I'm mainly in charge of the analysis of omics data in MECFS.
Thank you. So Dr. Lipkin, we heard briefly of some of the more recent exciting findings coming out of your group. And I want to ask a few questions around some of those findings that you presented. First, I'll begin with some of the energy metabolism studies. How would the metabolic findings refute the paradigm that depicts MECFS as a psychological disorder?
You know, that particular depiction of MECFS is so unfortunate. It actually goes back decades. My first engagement in this field was in the mid-1990s when we were asked to look at whether or not a virus called Borna might be implicated. And in those days, we went back, we used PCR, we used antibody tests, and we found no evidence for Borna virus infection. But I was impressed at that point, as were my colleagues, by the observation that people with MECFS in the Swedish cohort had very high levels of antibodies to a wide range of things, some of them that you don't even encounter in nature. So at the tail end of our paper, we said, we find no evidence that this particular virus is important in MECFS, but we do find evidence of abnormalities in the immune system, suggesting that this is a biologically based disorder. Now, several years later, when we received some support from Glenn Hutchins, the Hutchins Family Foundation, we created the Chronic Fatigue Initiative. And at that point, we began looking at cytokine abnormalities. And in pioneering work that was done by several members of the team, including Mady Hornig, who's not on the call today, we demonstrated that there was evidence of inflammation in people with MECFS. It was higher in people who'd had disease for less than 3 years. It began to diminish thereafter. We also found abnormalities in spinal fluid, also suggesting inflammation. So even before we did the metabolomic work, there was evidence that there were problems with the immune system. And as you'll hear throughout the course of the conversation today, particularly some of the work that Jason and Brent will be discussing, you'll see that there is a linkage between the abnormalities in the immune system and some of the metabolic disturbances that we've seen. But if you take together the immunological abnormalities and the metabolomic disturbances that we've seen, it now becomes very clear that people who want to say that this is a psychological disorder really don't have strong grounds whatsoever. So we believe this is extremely important in demonstrating that these are real illnesses and that people are deserving of medical solutions.
Thank you. Have you done studies examining the effect of cognitive exertion on oxidative stress or mitochondrial function in people with MECFS?
We have not done any work of that sort as yet, except that one of our colleagues at the Bateman Horn Center, of course, Cindy Bateman and Suzanne Vernon, have done these NASA lean tests where they show that there are cognitive abnormalities that occur when people get into an upright position. And we hope that as we begin to look at samples collected from those individuals after this lean test, that we will find that there will be abnormalities there. Now, I will say that the fact that we have abnormalities in the spinal fluid that we've examined, and Jason was involved with some of those analyses, confirms that in fact that there are immunological perturbations in the central nervous system. And these dovetail very nicely with some work that's come out of Cornell and their particular center, where they've shown evidence of inflammation using brain imaging.
Okay. Does your finding that inflammatory responses are not increased following the physical exertion challenge suggest that inflammation is not responsible for the symptoms of PEM?
Well, let me turn that particular point over to Jason. Jason?
Yeah, thanks, Ian. So to address that question, actually, that's not entirely true, because what we found was that inflammatory responses in PBMC to different microbial stimuli following the physical exertion were not increased in MECFS. And that is mainly because that the PBMC in these MECFS patients were already stimulated. I mean, they were maxed out, so that exertion will not possibly further induce inflammatory responses in these patients. So that actually showed how systematically ill these MECFS patients are. And actually, we have not looked into how physical exertion directly impact the inflammatory responses.
Okay, thank you for clarifying. How does an impaired TCA cycle and citrate buildup that you observed after exertion relate to the symptoms of PEM?
I think I'll address that question too. So we found that the levels of citric acid in healthy controls significantly decreased from pre to post physical exertion, while the citric acid levels in MECFS patients marginally increased from baseline to post exercise. So the reduction in the controls indicate that healthy people are utilizing the citrate in TCA cycle to generate ATP molecules and produce energies. Well, however, in MECFS patients, the accumulation of citrate suggests that there's an impairment in the TCA cycle such that they cannot generate enough ADP molecules for these patients, which can at least partially explain the symptoms of PEMs in these patients.
Let me add just one thing there, which I think is helpful for Rochelle for putting this into perspective. The original work that came out of the Navio team at UCSD was really trailblazing and demonstrating abnormalities related to the mitochondria. So what we've done is to extend and build on that by looking at mitochondrial function. So it's entirely consistent with what was reported there. And again, it brings in this potential link to the immune system, which attacks these mitochondria and causes some of this damage.
Okay, thank you. Next, we have a few questions relating to the microbiome studies. First, what are some of the theories of what causes the microbiome changes that you observed? So you observed increased overall bacterial populations, but a decrease in butyrate producing populations.
So that's for Brent, I'm sure.
Yes, so, and you've listed sort of several theories here that potentially could explain these differences that we found in the microbiome, including chronic inflammation, exposure to antibiotics, differences in the virome, inherent differences in gut immunology, sedentary life, or a low carbohydrate diet. And firstly, I would say that to some extent, these theories may not be independent theories. But rather that the complex interplay sort of among these factors could contribute to the microbiome changes in MECFS. But that being said, I would suggest that there are two theories that I think have some relatively strong evidence to support them based on the literature. The first theory relates to acute viral infections rather than the virome per se. Increasing evidence has shown the potential for acute viral infections to mediate changes in the gut microbiome, and this is particularly the case for respiratory infections. And those include influenza, RSV, and COVID-19. And all of these viral infections have actually been shown to affect the gut microbiome. And in fact, all have been shown to reduce butyrate producing bacteria. So you could have sort of an initial acute viral infection that starts off this initial alteration of the microbiome. What we don't have good evidence for is how long these changes associated with an acute infection may last. So COVID-19, I think, is providing some good clues in terms of in that respect, and a recent paper published in Gastroenterology basically showed that some of the same bacteria that we're seeing deficient in MECFS patients are deficient during infection and during the post-recovery period. But we still don't have good evidence to how long that can last. The second theory, could also dovetail onto the first theory in terms of, what maintains that deficiency in butyrate, that initial deficiency in butyrate. And that could relate to sedentary behaviors. A lot of studies in exercise physiology have shown that physical activity increases levels of butyrate producing bacteria, and individuals who are sedentary have lower levels of these bacteria. So there may be an initial infection that then is even becomes more pronounced differences in the microbiome that relate to sedentary behaviors. And then there are other aspects in terms of how chronic inflammation could be set up, or changes in inflammation in the gut that relate to these deficiencies in butyrate, or even in the overgrowth of bacteria in the gastrointestinal tract. So I think that many of these factors could be contributing to something that is an initial sort of instigator of the disease.
Okay, interesting. How will you go about determining if low butyrate producing bacteria may be a cause or consequence in MECFS, both for the disease as a whole, but also for particular symptomology?
So I think that the best way to go about that would be to conduct a randomized clinical trial in MECFS subjects to look at how probiotics or prebiotics or a combination of the two, symbiotics, are able to alter butyrate producing bacteria, to boost the levels of butyrate producing bacteria and butyrate itself, and whether that has any impact in terms of alleviating symptoms associated with MECFS.
Okay, and are the butyrate findings clear enough to potentially be a biomarker? You touched on this in your previous answer about finding the similar decrease in butyrate producing populations in the cases of other acute viral infections, especially respiratory diseases. And we were also curious whether this is specific to MECFS and if its specificity is sufficient to serve as a biomarker. It sounds like that's not the case.
So as an independent biomarker, I would say probably not, but in combination with other biomarkers, it may add support. So if you have deficiencies in butyrate in combination with specific metabolite changes, then it may be able to sort of enhance the predictive power of biomarkers in combination. But in addition to the viral infections that I've mentioned, deficiencies in butyrate have also been associated with other diseases, human diseases, including inflammatory bowel disease, colon cancer, obesity, metabolic diseases, including diabetes. So I think as a specific biomarker, it's unlikely to be a great biomarker for.
Sure. Nevertheless, exciting for a potential therapeutic intervention if those trials are successful. All right, thank you. Next, I'd like to ask some questions about some of the immunology studies that you've done. How will antibody tests advance your understanding of the hit and run hypothesis?
I'll take that, Rochelle. So we've looked using molecular tests that allow us to find genetic material associated with bacteria and viruses to characterize those, to define differences in people who have disease, who don't have disease, who have severe disease, all of those sorts of issues. I think we've really tried to push that as hard as we can in the first 5 years of the work that we've done as a center. During the second phase of the research we're doing, we're going to try to find evidence for infection that may no longer be present at the time that we make the diagnosis. Our notion here is that somebody has an infection that triggers damage that results in the onset of autoimmunity or some sort of metabolic disturbance that perpetuates itself. And the way in which we do this is by looking for antibodies that are specific for bacteria and viruses. We've done this now for many years in other contexts. We did it, for example, with a virus called enterovirus D68. We're able to use these kinds of methods to demonstrate that kids who had a polio-like syndrome, where we could not find nucleic acid, nonetheless had evidence of infection with enterovirus D68. If something similar could be identified in the context of MECFS and even a small subset of people, you might be able to get at vaccines that could prevent the onset of these sorts of diseases. And some of you may have read in the past couple of days that there's a suggestion that Epstein-Barr virus might be implicated in multiple sclerosis. So our hope, of course, is that we find something like that. Because we don't know what virus we're really looking for, we have to use very broad methods that look at all viruses and all bacteria. So we're building those kinds of platforms for doing that sort of work now.
Okay. Have you looked for potential correlations with onset type or particular symptoms that correlate with the B-cell clonal expansion you've observed?
We haven't done anything of that sort yet. The B-cell clonal expansion suggests to us that there is some stimulus that is eliciting a very specific type of change in the way B cells that make antibodies grow, replicate, and become invested in targeting specific antigens. One of the things that we can do in the future is to try to identify specific populations of B cells and try to identify their specific targets. Our focus always has been, in the context of the MECFS program we run here, to focus on people who have a history of some sort of a disorder that sounds like it's infectious prior to the onset of MECFS. We realize that there are probably multiple triggers for MECFS, but we think this is probably the lowest hanging fruit if we're going to try to find something that gives us insights. Now, of course, with the onset of the COVID pandemic at PASC, we've been slowed in our ability to do some of our MECFS research. The good news, if you can call it that, is that there's an opportunity to look at a recent well-characterized viral infection and begin to understand how it might result in something that looks like MECFS. And there are many people who have PASC who look very much like they have MECFS. And Tony Komarov and I wrote a review article on this a while back that I'm sure you've seen.
Yeah. All right, thank you. Is there a correlation between baseline and post-exercise IL-6 levels with disease or PEM severity? So this is the exercise challenge that showed high baseline IL-6 even prior to exercise, you know, indicating these people already have some inflammation at baseline. Does that correlate with their disease severity or their symptom severity?
Very, very good question. I don't think we have evidence on that yet. I would just point back to what Jason said a few minutes ago, and that was that most of the work that we've done thus far with the exercise challenge has been to look at the effect of exercise or not on triggering some additional damage in terms of these B cells peripheral blood mononuclear cells that would produce various immune markers. And there, what he said, and I agree, is that it looks like even at baseline, people are so stimulated and have such cytokine responses that in fact, they may not have any, any residual ability to respond. This could lead to increased susceptibility to infections, lack of resilience, a whole range of things, which will sound very familiar to people with MECFS. Mm-hmm. So everything looks to us as though at baseline, for whatever reason, people with MECFS seem to be more sensitive to a wide variety of environmental stimuli. This revs up their immune systems and has an impact on them metabolically as well.
Okay, thank you. You mentioned estrogen's effect on this disease. Have you seen anything regarding estrogen-progesterone balance or suggestions of progesteronic interventions as treatments for MECFS?
Well, Jason, do you want to talk about the impact of estrogen and androgens, for that matter, on innate immune responses?
Yes, I can describe the data briefly. So what we found was the cytokine levels in PBMC in response to Staphylococcus superantigen stimuli has a dramatic difference in sex as well as in age. So we found that more pronounced elevations at baseline before the exercise of elevated levels of pro-inflammatory cytokines in MECFS patients, but mostly these elevations are more pronounced in female subjects. Particularly within the female subjects, we studied whether sex hormones or sex steroids have an impact on these elevations. We separated by age of 45. I found that these elevations are much higher, so we have an extremely high response in the female patients older than 45, with lower levels of estrogens. And because the estradiol can regulate the pro-inflammatory cytokines, with lower levels of estrogens do not have that capacity with the MECFS of this regulation that leads to the extreme elevation in pro-inflammatory cytokines.
Okay. Next, I have a few general questions that could apply to any of the findings that you presented. Do any of the measures analyzed correlate with subjective disease severity or disease duration, something that you've examined in the past?
Jason, you want to talk about our symptom scores?
Yes. So based on our analysis in the sex and age stratified fashion, within each of these subgroups, the number of subjects become extremely low, even in the female subjects. We have tried to correlate the levels of pro-inflammatory cytokines with severity of symptoms. But because the number of samples is low, that possibly the correlation coefficient could be biased. So I'm not, we don't want to draw the immediate conclusion yet without confirming it in a larger study.
Sure. Okay, thank you. Has your research included study of comorbidities to try to classify patient subgroups?
Well,
Yes.
we've looked at people who have disease for less than 3 years or more than 3 years, which I think is, there's some clear differences in cytokine responses in those individuals. It almost looks like after 3 years, people begin to burn out and their ability to mount immune responses. We've also looked at people who report GI comorbidities, people who have self-reported irritable bowel syndrome. They don't meet full criteria necessarily for irritable bowel syndrome, but there are dramatic differences there that I think Brent has found in the microbiome and Jason has found with respect to metabolomics. So Brent, would you like to talk a little bit about IBS?
Yes, so essentially we in looking at the microbiome data, we could sort of immediately see, based on sort of general measures of the microbiome alpha diversity, that there were specific changes that seemed to be occurring in individuals who had MECFS with IBS. They had reduced diversity of the microbiome, whereas MECFS patients without a diagnosis of IBS did not have those changes in alpha diversity. But most of the changes that we found in the microbiome were independent of IBS comorbidity. So there are specific changes in the microbiome that do occur in individuals and subgroups that do have IBS. But overall, there are major changes in the microbiome just associated with MECFS.
Jason?
So in terms of the metabolomics and cytokine responses, we did not find a IBS specific disturbances in these assays. Again, mainly because there are less than optimal number of IBS patients within our cohort. So in generally, when we focus on the stratified analysis in MECFS patients without the irritable bowel syndrome, compared to all the controls without irritable bowel syndrome, the findings are quite consistent with the overall population in our cohort.
Okay.
But in terms of other not necessarily comorbidities, but sex and age, that's showing very distinct patterns.
Okay. Well, that gets to my last question, which is, have you conducted the AI analysis of symptoms to define subgroups within the MECFS population? Similar to the diagram that you showed, the Venn diagram overlapping Gulf War and post-COVID with MECFS. Do you see clusters of symptoms, for example, with onset type, or you've done the studies on disease duration? Have you kind of leveraged that technology to parse just that MECFS population?
So Jason, we remember this, we had that diagram in the paper where we looked at symptoms, pain, for example, cognitive dysfunction, so forth, in association with specific metabolomic findings. I think Jason has talked about the challenge before Rochelle, where we don't have enough subjects to find something that is statistically significant. But there were some trends which you might be able to describe, Jason.
So in terms of the identification of subgroups, I think this is actually my immediate future plan for integrating multi-omics data in MECFS to identify particular endophenotypes, both in the plasma as well as in the PBMC of the supernatant samples. We have found evidence in the proteomics, in the metabolomics, in the cytokine immune response and everything. Once we integrate all of them together using a machine learning technique, the strategy I have planned is to derive similarity matrix within each of the omics data, and then we can fusion them together using some machine learning techniques, and then run another clustering analysis. Maybe we can find something unique in that, in contrary to focusing on one particular omics data.
Okay, thank you. It sounds like the cohort size is the major challenge in that approach. I want to thank you all for your time this morning, and I want to thank you sincerely for your work in studying this disease on behalf of millions of patients around the world. We are grateful for what you're doing, and we are excited to see these objective results reported that prove the severity of our disease. So thank you very much.
Rochelle, may I just add one thing?
Mm-hmm.
So First of all, I just want to highlight the fact that you've seen two people who are not working in MECFS before the center was established, who are turning their talents and their intellect to trying to address the problems and get clarity. We have at least another 8 people who were not represented here today who could make similar kinds of contributions. And if you replicate that across the 3 centers, there's an enormous amount of talent working on this particular problem. And I'm cautiously optimistic that in the next installment of these centers, we're going to be able to move into solutions, which is why we started this in the first place.
Fantastic.
Thank you, and for your belief in us.
Thank you. Thanks a lot.
Thank you.